We will move on to the second talk. We'll have the question and answer sessions in the end of the session. Inflammatory bowel disease is something which is very little understood. There is like confusion about the etiology. Management of inflammatory bowel disease is something which varies from country to country. Our country probably follows a different pattern. There are new drugs coming in. But the most exciting part is about fecal microbiota transplant. And we have with us Professor Vineet Ahuja, who's a professor of gastroenterology at All India Institute of Medical Sciences. He's also the Associate Dean of Clinical Research, and he's a very dear friend. So he will be highlighting the role of fecal microbiota transplant in inflammatory bowel disease. Professor Vineet Ahuja.
Thank you, chairpersons. Good morning, everyone. First of all, I'm grateful to Professor Ganguly and Dr. Nija Ajela for giving me the opportunity to come out here. And I would also like to congratulate Professor Ganguly for what I would call like science stewardship or leadership in terms of getting all the young minds out there and continuing to enthuse them by getting them exposed to the best in science. Thank you so much, sir, for this great initiative, sir. I'll be talking about microbiome manipulation. Sorry. Okay, thank you. So I'll be talking about microbiome manipulation therapies in IBD, and we have had some very exciting talks since morning, and every talk has taught us so much. Now I'll start with an example out here. For example, just to tell you that how science uncovers certain things and how things which seem to be of common sense may not be true. Like what are these? These are bears. Like you had a great talk by Dr. Yajnik, then you had a wonderful talk by Professor Chandy, and so if you were to, as students of science, if you were to take bear as an animal model, what kind of disease would you take the bear as animal model for? Diabetes, fatty liver, something like that, metabolic syndrome. You would all agree? All of you would agree for that? Yes or no? Yes. Well, bears don't get diabetes. In fact, recent research shows that hibernating bears could hold a clue to treating diabetes. They probably have 8 key proteins which have been found that they hibernate. They have great BMI and etc., but they never develop metabolic syndrome and diabetes. So this is how baffling things can be. And inflammatory bowel disease is another example of a disease which can be very baffling. It is a disease of unknown etiology, chronic immunoinflammatory disease comprising of ulcerative colitis and Crohn's disease, where you get ulcers in the small intestine or the large intestine. It affects people in the prime of the youth and unfortunately stays with them all through their life. And as yet, we have not been able to find a cure for inflammatory bowel disease. A lot of research has gone into this field. And if you look back over the last 2 decades, you find a plethora of drugs which have come up. Monoclonal antibodies, oral small molecules, anti-TNF agents, lymphocyte trafficking inhibitors, IL-23 inhibitors, IL-12/23 inhibitors, JAK inhibitors. And yet at the same time, we find that all these drugs, they have what we call as a therapeutic ceiling. They act, but they lose their response soon after. They don't act in more than 30%, 50%, or 60% of cases. You don't get 100% of response, and at the same time, they won't, till the time patients are taking these drugs, the patients have a response. The moment the patients stop taking these drugs, majority of them lose their response. So obviously, there's a big issue out here. So why does it happen? Like in the sense that why? Because most of these drugs, they are acting on their immune modifying, and we knew that there is a lot of redundancy in immune circuits. So probably that if you're blocking one hub, the immune mechanisms are going through another hub and again exciting inflammation. So if you go back to the drawing board, what you would do, you would go back to the pathogenesis of disease and see whether you can act on it or in a different possible manner. If you look at possible culprits for inflammatory bowel disease, we find that there's a genetic susceptibility, there may be altered epithelial cell responses, aberrant immune responses, and again, another very important component, that is environmental factors, of which diet and gut microbiome could be a principal player. So do we have a gap in knowledge out here? Can we attack the microbiome out here? And can it change the course of the disease? That is the question. So we obviously have 4 generations of gut microbiome manipulation. We have the antibiotics, the diets, the prebiotics, the probiotics, fecal microbial transplantation, and also postbiotics, as Dr. Chandy showed you in a slide. Postbiotics are also coming up. So what is fecal microbiota transplantation? It is essentially a restorative approach. In probiotics, you're giving one strain, some billion spores and all. In FMT, you're throwing everything in, all the bacteria, all the viruses, all the fungi, all the archaea, and it is infusion of a healthy suspension from a healthy individual into the GI tract of an individual with the disease, with the hope that it would refurbish the microbiome out there. And if it refurbishes the microbiome, it resets the immune system and possibly changes the course of the disease. So I'm not going to show you any animal experiments, mice experiments. I'll just show you a simple real life animal experiment, in the sense that this is published in Gut, which is a top rated journal in gastroenterology circles. So this is kangaroos. Kangaroos, they have small babies, which are known as zoes. So zoes, when they get orphaned, because if the mother kangaroo is poached and all, these zoes are often exposed to conditions which predisposes them to diarrhea, etc. So Cynthia Ranch, who was a game rancher, what she did was that she took up the poop from healthy kangaroo babies, and subsequently she made what are known as the microbiota poo shades, and does the video run? Okay, if it doesn't run, I'll leave it. Yes, it runs fine. So what I did was that she took healthy zoes, took their poo, that is the fecal matter, mixed them with milk, lactose-free milk, and fed it to the young kangaroo babies who were very sick, who had a lot of diarrhea. And she had thousands such cases where all of them improved, clearly showing that there is some wisdom in this approach, and this can work. So now let me come to the first question, that before you're going to change the gut microbiome, you have to first decide whether it is the cause or the effect. Previous speakers have talked about it, and I've told you that possibly we are not very clear whether it is a cause. Let's see, in IBD, in inflammatory bowel disease, if you look at gut microbiome changes, you find that compared to other healthy adults, or compared to diseases like amoebic colitis, there's a different kind of microbiome which is seen in inflammatory bowel disease. This pattern was seen in North India. This pattern is also seen in South India by CMC in CMC Vellore by Professor Ramakrishna, who have shown, yes, there is a distinct signature in patients with inflammatory bowel disease. But that does not answer the question whether it is a cause or effect. Similarly, in inflammatory bowel disease, you may have patients who are in remission, mild activity, patients who are very sick, and you look at the gut microbiome signatures, you find that there's a difference in gut microbiome signature in patients who are doing well compared to patients who are very sick or who have very severe disease. But again, this could just well be the effect. Then what we also picked up was we picked up healthy people. Let's say people who are living in rural areas, people who are living in urban areas, then people who are living in Ladakh, because that is a place where we have set up health camp every year, and we have never seen a case of inflammatory bowel disease out there from last 2 decades, just to look at the gut microbiome. And what was surprising was that urban people, they had a worse microbiome as compared to rural people or the lay people. So if you collect all this information together, you find there's a straightaway. Acute severe ulcerative colitis, worst microbiome. Ulcerative colitis, slightly better than them. Urban healthy, normal, but inferior to rural healthy. Rural healthy is slightly inferior to high altitude healthy. Again, it does not show that there is a cause or an effect, but it tells you there's a lot of association out there. And if there is a lot of association out here, it seems reasonable to manipulate the microbiome. So comes up fecal microbial transplantation, where essentially you take donor feces, mix them, blend them, filter them, and with the help of endoscopy or oral capsules, you infuse them into the person with GI disease. We often use most of the times we use a colonoscopic approach, but sometimes in patients, particularly with liver disease, what has been seen that the upper GI approach, or putting the putting it into the duodenum, which is the first part of the small intestine also, you can do fecal microbiota transplantation. A component of this is a donor, and donor needs to be very, very healthy. It is not healthy in the sense, in the regular sense that, okay, I'm a healthy person. Now, the donor has to be extensively screened because you may end up transmitting some disease. So the donor has to go through a very rigorous questionnaire in terms of travel history, in terms of infectious history, in terms of drug history, in terms of various exposures, and also various kinds of testing, so as to be very sure that the donor is healthy. In fact, even if somebody is healthy but has a BMI more than 25, we do not take up that person as a donor for fecal microbiota transplantation for the theoretical risk that you may end up transmitting metabolic disease, etc. So these are, if the videos work, these are the ways we do fecal microbiota transplantation at our center. We have been doing it since 2014. And FMT is not restricted only to GI diseases. It has been seen in various non-GI diseases also. A lot of data is coming up where it has been shown that it is effective in autism, Parkinsonism, schizophrenia, Alzheimer's, even in augmenting immunotherapy for cancer responses. So it has a whole lot of audience. It is something like which is probably becoming a movement. So I come to my next part of the talk now, where I would tell you about our experiences in terms of changing the microbiome. And so, microbiome manipulation can be done with the help of fecal microbiota transplantation. It can also obviously be done with the help of diet, because diet would change the microbiome. So the research hypothesis was that in patients with ulcerative colitis, which I told you is a kind of inflammatory bowel disease, if you do FMT, along with that, you give the different kind of diet, an anti-inflammatory diet. Can you induce remission? And subsequently, for the next 1 year, can you maintain the remission? So we did a randomized control trial and published it, which was fecal microbiota transplantation with anti-inflammatory diet, followed by anti-inflammatory diet alone. And it was found to be effective in inducing and maintaining remission over 1 year in patients with mild to moderate ulcerative colitis. Essentially, the trial design was that patients of ulcerative colitis were randomized into two groups. One group continued the standard medical therapy. These were patients with mild to moderate disease activity. The other group, 7 sessions of fecal microbiota transplantation were given over an 8-week period. Along with that, the diet was also changed. They were given anti-inflammatory diet. After 8 weeks of FMT, anti-inflammatory diet was continued for a period of 1 year, and we looked at clinical responses, looked at endoscopic or mucosal healing, and also differences in fecal calprotectin, which is a marker of inflammation. The anti-inflammatory diet which we gave, the principles were to avoid lactose. Yes, you can give curd and buttermilk, to avoid gluten-based grains, to avoid processed food, canned food, red meat, and encourage plenty of fresh fruits and vegetables. The clinical outcomes showed that at 8 weeks, as well as at 48 weeks, there was a significantly better response in patients who were given FMT and anti-inflammatory diet. And even the endoscopic responses, that is mucosal healing, was superior in patients who were given this kind of therapy, suggesting that this is proof of concept. In a way, this therapy does work. Obviously, it needs to be validated in much larger trials also. And the interesting thing was there was no major adverse event. And secondly, obviously, if you're going to do a cost-effective analysis, it is going to be much cheaper than giving the monoclonal antibodies, where the cost can range from 2.5 lakhs to 6 lakh rupees a year. So the next part is that Professor Chandy showed you that, okay, you have a concept. Subsequently, you would like to characterize it, do a compositional characterization, functional characterization, then build up a hypothesis, then take it to another animal model, test it out, and then go to postbiotics. Let me see that if we can complete the story out here. So these are my 2 PhD students, Manaswani and Aditya, who have done this very nice work, where all these patients who received FMT, they took the samples, the fecal samples, as well as mucosal samples from them, looked at the gut microbiome and also the fungus, the gut microbiome, and also looked at metabolites, because metabolites are more important than microbiome. And also looked at the same patients when they had got a response to FMT, what happened to the microbiome, and metabolome. And as I told you, they looked at 2 sites, the mucosa-associated microbiota as well as the fecal microbiota. So at both the sites, I'm not going to go through the bacteria, but just to show you broadly, yellow is the UC circle, that is pre-FMT. Pink is the post-FMT. See the diversity, richness, composition, how it has changed. and it has become so much near the controls. And it has happened at both sides, both at the mucosa as well as the fecal. Similarly, we see similar kind of trend with metabolites. Metabolites seem to normalize, move towards a level of controls, the kind of composition of metabolites which control population had after patients had received FMT. Again, you see fecal metabolites as well as colonic tissue metabolites showing a very good response after FMT. So this is concluded that yes, there is a change, that is a possible mechanism. There's a change in bacteria. There's also a change in the metabolite that helps. So the next step was that you're showing it at fecal level as well as mucosal level. Is it possible that there's another niche which is more conserved and possibly that is a master regulator? Like stem cells are the master regulators. It's like somebody who's close to the prime minister possibly has more chances to influence, something like that. So there is another population out there which is known as the crypt out here. Do crypt have bacteria? Do those bacteria possibly have a more influence? So the next part of the study was that fecal bacteria have been looked at, mucosal bacteria have been looked at. Let's look at crypt bacteria. And so it took a lot of effort to get those bacteria from there because they're very sparse in number. You have to do fish. Thereafter, you have to do laser capture microdissection, and thereafter do the 16S. And clearly, again, it showed that even crypt-associated microbacteria, there has a completely different population. They're more aerobes. They are not direct facultative anaerobes or obligate anaerobes, because obviously the oxygen concentration is higher out there. There are aerobes out there, and those aerobes also, there was a different population in patients with ulcerative colitis, which changed after fecal microbiota transplantation, showing that yes, the benefit comes out there also. So the mechanisms were there's a change in microbiota and microbiome at fecal level, mucosal level. There's a change in microbiota derived metabolites following intervention at fecal level as well as mucosal level. And there's also a change in crypt specific microbiome following intervention. We couldn't do the metabolites at crypt because of the very sparse material which comes out from there. So this part of the story is completed. You've done a clinical trial showing that in a disease, ulcerative colitis, randomized clinical trial showing the benefit of FMT and anti-inflammatory diet, establishing FMT as a therapy. Thereafter, you have looked at microbiome changes, metabolite changes at 3 sites, and of this, from the heat maps, we picked up a very promising signal for urolithin. Urolithin, which is found in pomegranates, etc., and it was seen that it was very much increased after FMT, particularly in patients who are responding. So obviously, the next part of the trial was that, okay, the hypothesis has been built up. And as Dr. Chandy showed you, now is the time to go back to animal studies. So what kind of model are we going to build up out here? We decided let's look at the big organoid lab, and in organoids, test out whether these metabolites work or that. Organoids, as you all know, are 3D culture systems. So again, instead of mice model where obviously the genes would be different, here these are patient-derived organoids where you take biopsies from colonic biopsies and subsequently you make the 3D spheres of them, spheroid cultures. And so organoids were set up from non-IBD controls as well as ulcerative colitis patients. The organoids were treated with urolithin, which is a metabolite, for interest for us, so as to look for the changes. And for changes, we did single cell transcriptomics to find out whether there is any decrease in inflammation or not, what happens in various cell populations. So these are the various organoids which are growing, and they were all enriched in the intestinal stem cell population. Subsequently, urolithin was added to them, and it was seen that with the help of urolithin, when single cell transcriptomics was done, essentially what was seen was there was a decrease in the inflammatory population, which was very much present out there in patients who were treated with urolithin, patients of ulcerative colitis who were treated with urolithin. So all the three processes have been completed till now, and now we are looking at forming postbiotics in terms of looking at urolithin-based therapies and how we can go and in terms of making a capsule out of them, capsulized form of them, and whether that can be useful or not, or making a cocktail of metabolites and finding that it is useful or not. And finally, another interesting point out here, this is for all the people sitting out here who want to work in this field. There's one question which is absolutely unanswered out here, and whoever answers this question obviously is going to make a major contribution to this field. And that is the single most important factor in all of this is that some donors somehow have a profound effect in terms of getting the success for the endpoints. In terms of some donors have a profound effect on the microbiome when for the diseased population. So these are what we call as super donors. So how do you identify the super donors? What kind of microbiome population do they have? What kind of diversity do they have? And I think that's a key question which needs to be answered. And finally, just two slides I have, since this is in Kerala, and there's a recent study which came out from our group, and this is essentially a coconut water. This is a dietary change, and how simple things can influence the microbiome, can influence the disease activity. It is not always that you need to go in for most expensive things. So this trial showed that coconut water was given to patients with mild to moderate ulcerative colitis, and it reduced clinical remission. It did not induce endoscopic remission. So in the sense that it gave a marginal benefit, not a very major benefit as you would expect with steroids, but at the same time, again, something which can be easily practiced. It was a well-conducted trial, a rigorous trial in terms of it was a randomized, blinded, placebo-controlled trial where patients of mild to moderate ulcerative colitis were given for 8 weeks, 400 ml of coconut water was given. Why? Because we thought it's a good source of potassium, and previous data from our group and THSA, Dr. Amit Awasthi's group had worked on it and shown in mice model that when you provide potassium, it decreases inflammation as compared to sodium, which increases inflammation. So when you provide a source of potassium, you give coconut water for 8 weeks. So it is like adding a good dietary thing, the dietary addendum. It was seen the patients achieved clinical remission, clinical response, and fecal calprotectin also fell down. Endoscopic responses were not seen as were seen with fecal microbiota transplantation. Interestingly, there were a lot of changes in microbiome, which we also saw when patients were given coconut water. So this is what I would say proof of concept study. Again, obviously requires validation in a larger trial, but provides first of its proof, which shows that 8 weeks clinical remission and response was significantly higher in patients who received coconut water compared to placebo. And coconut water increased the relative abundance of bacterial genera, which negatively correlated with clinical and endoscopic disease activity in patients with mild to moderate ulcerative colitis. With this, I end my talk, and thank you so much. This is our IBD group, and these are collaborators from THST, RCB, and ICG. Thank you so much.
Thank you. Thank you, Dr. Vineet. I think you have really stimulated the young minds here, and we are extremely thankful for you to have shared your elegant, detailed research in such a meticulous manner. And I'm sure all of us, including me and many of us sitting in the audience, have a lot of questions which we will take after the next presentations. Thank you very much.